experimental work here. And today I'm going to present results from a grant that we were awarded last July, and I'm very, very much excited about this project, and I hope you find the results interesting. This is very much ongoing work, so some results are still preliminary, and this is from a joint project with Francesca Luca at Wayne State University and with William Wan at University of Michigan. So one of the things that I would like to start as introducing is why the environment is important. And one of the reasons is as humans got out of Africa, they had to adapt to radically new environments. So they had to change the diet to new foods. They may have had to adapt to a colder climate, maybe less sun exposure, different set of pathogens. So they had to adapt, and many of these adaptations consisted in natural selection of advantageous genotypes, and different genotypes were advantageous at different environments. Today, perhaps some of the adaptations are not useful anymore, and perhaps gene-environment interactions can explain some complex disease traits in humans, and perhaps explain part of that missing heritability in genome-wide association studies that Nancy Cox was talking before. So as she was saying, now it's more recognized that complex traits and diseases are usually influenced by hundreds or perhaps even thousands of genetic locations. And some of these locations might have different influence on the disease trait under different environments. So for example, I mean, maybe some people might respond to a treatment and others not. And that's also one of the things that we still have to investigate. So what is the environment, or what is our definition of environment? In one sense, will be the organismal environment, which can be very complicated. It's multivariate and it's hard to control in genome-wide association studies or in epidemiology studies. And you might be exposed to a combinatorial of many different agents, and you might exhibit many different types. What we could do is what we would like to do is maybe to use develop an in vitro system where we could test all these different environments in a more controlled manner, where we could take different cell types, treat them with one compound at a time, and measure some molecular phenotypes, and then do a QTL analysis, and then use this QTL information to inform the GWAS studies. So our goal here is to go to many environments, and for this we would like to identify which genes have genetic variants that may have different response to different environments. And to do this, we would like to develop an efficient experimental and computational approach to screen potentially hundreds of different environments. One tool that has been used for doing this, that it's very successful, is also a QTL approach. And basically what you do here in terms of, it's map response QTL. So compared to what you do in the GTEx projects, that you have different tissues, in this case you will repeat the QTL on cells that they are in the control, that they don't have a treatment, and in this case you do the same individuals. We treat them, for example, with glucocorticoids. This is from one of the first studies that it was done this, and you can see that for this gene there is almost no change in gene expression with the genotype, and for this one there is an increase in gene expression with the minor allele. There are a few of these studies, but it's very limited compared to the amount of potential environmental exposures that might exist. And also for an EQTL, you need a large number of individuals to have enough power. So in conclusion, we need a different approach. And here it's what we proposed to do. So we developed an approach that for high throughput and cost-effective identification of condition-specific gene and bioenvironment QTL genes, and it has 2 steps. In the first step, we use a shallow sequencing to identify distinct gene expression profiles across many different cell types and treatments. Because also prior literature, sometimes you don't know which cells, which treatments. So basically, we do it in a highly multiplexed way. We get 3 individuals at a time, and then we do a panel of treatments and across different cell types. We multiply 96 by 96, and then we will sequence each library 4 to 5 million. And this is enough to distinguish if the cells have changes or not due to the treatment. And then for the second step, we use a selected number of these libraries, we deep sequence, and then we use an allele-specific expression approach to identify which genes have genetic variants that interact with the environment. Which treatments we use? Basically, we included panels that target either dietary components that might reflect the environmental pressures depending on which food was available, hormones that might mediate response to stress, for example, glucocorticoids, hormones that represent natural physiological or pathological changes like insulin or estrogen. Also, we did over-the-counter drugs like aspirin and ibuprofen and allergy treatments, and also some environmental contaminants and common chemicals. So for example, things coming from plastic leaching, BPA, phthalates, and that probably are much recent during the human evolution and probably we don't have time to adapt. And also, we do water treatment byproducts and also polluted air from Detroit. And then which cell types do we use? In this case, we use primary cells because we need to grow the cells and do the treatments. And in this case, we use primary cells isolated from the umbilical cord. In this case, UVEX and smooth muscle cells. And also we use PBMCs isolated from whole blood, and also lymphoblastoid cell lines because that they are divided from B cells, and the reason we do is because they are fully genotyped, and also melanocytes. The reason we pick those ones is because they are quite well characterized by the ENCODE and roadmap genome, so we have a lot of information that then we will be able to integrate with. And combining together the treatments and the cell types in total, in this case, we will monitor about 250 cellular environments. That with 3 individuals per each cellular environment, we prepare a total of 1,000 RNA-seq libraries. We sequenced them. We did DE-seq to identify gene expression. And here highlighted in red will be the cellular environments that had more than 100 genes different than the control. And those ones will be the ones that we select for the next step. We can see that things like vitamin A, retinoic acid, you have gene expression response in all the cell types. But other things like selenium, maybe you only have two cell types, and for zinc, maybe only 1. So basically, we selected these cellular environments, and then we do deep sequencing. And here is also to visually describe the data, and we show a principal component analysis prop from the treatments for a subset of the treatments that we selected for UVEX. On the first PC, we can see we can see that the 3 individuals that we have for each treatment tend to cluster very well together. And the first species will separate this metal ion from the rest. And the second principal component will separate these vitamins and DX that they bind to nuclear ligands, so they have a different biology that what my caffeine might have. And for things that even for retinoic acid that we have respond across all the different cell types, we can see that there is also clustering in cell-specific responses. So the idea is that you also have a cell-specific response to the treatment because otherwise you will see all these points together. So basically, UVEX might have different gene expression response profiles than from melanocytes, for example. And then here we are finally on the second step, that bringing back the genetic variation. So we had the environmental perturbation, and now we want to examine the interaction with the naturally occurring genetic perturbation. And in this case, what we use is allele-specific expression. And for allele-specific expression, what you need is an heterozygote genotype at the body of the transcript, and then you will count reads, the ones that carry one of the alleles and the other one. And if there is, and then if there is an imbalance, if this is different than the that one-to-one ratio, this will be an indication that there is an EQTL, basically that there is a difference in gene expression with the genotype. To do this, we use an approach that we call quasar, that we recently developed. In this case, because we sequence the same individual across many conditions, we wanted to use joint genotyping and specific analysis using the same method. And with this approach, basically, we have a model that if you are an homozygote genotype, you can only observe these two reads, and with a probability of error if you do an error sequencing. And if you are in heterozygotes, you also have that, but you sample the reference or the alternate allele with this raw parameter that will be that under the null hypothesis, you will expect that it's 0.5, that you observe the same thing. And if not, you might see an imbalance. And when we apply this across all the conditions that we generated, this will be the QQ plot across all the conditions. And at an FDR of 10%, we detect about 9,000 unique genes with ASC that will be in any of the conditions that we measured. And the method is also available as a preprint on bioRxiv. And then the question still remains, which one of the genes have differences on the control versus treatment? And to answer that question, we use two parameters that they estimated in quasar. One will be this beta parameter, that it's the log odds for the reference allele and represents the allelic imbalance. And also we have an estimate of the standard error coming from the likelihood model of this parameter. And we have we have the estimate of the variance of that parameter for the control and the same for the treatment. And then we use these parameters and we fit it to MESH. And MESH, it's a Bayesian method for genetic association analysis with heterogeneous subgroups that was developed by William Wen. And basically uses the concept of configurations. And this has been also been used before, like in the GTEx project, and also in response to the environment. And basically, you assume that you can have four possible things going on here. First, that you might not have ASC in either condition. In this case, you will have all these beta parameters parameters equals to 0, and that will be the probability that that's the case. And then that you have ASC in one of the two conditions. And for this case, we use that the beta parameter is different than 0, or you can have only ASC in the treatment, and that will be equivalent that beta parameter of the treatment will be different than 0, and the other one equal to 0. And for the control only, the treatment will be 0, and the beta for the other one will be different than 0. And we do this for each control treatment pair. And what we get out of this is that most of the allele specific expression is shared for both conditions. But we wanted to investigate this a little bit more, and we wanted to see if this depended in the changes of the gene expression in response to the treatment. So in here, what we have in this plot, we partitioned all the genes in the log-fold change in the gene expression response that we obtained from just looking at differential gene expression of the gene. And then in the y-axis, you have the posterior density distribution of those parameters that indicate treatment only or control only. And you can see that as you have a higher fold change in gene expression, you are a little bit more likely to get condition-specific ASC. And here, this some of the examples that we obtained of from control-only ASC, and on the left, and for treatment-only ASC on the right. And basically, the ones that are control-only, basically, you don't see significant allelic imbalance on the control. On the treatment, but you see it on the control. And on the right, you see that the control, there is not significant allele-specific expression, and you see it for ASC. And in this case, are examples from different treatments: ibuprofen, vitamin D, dexamethasone, caffeine, and we still and this is very recent data that we got. And for example, this one. It's one of the first that we look, and it's in one gene, EPAS, that it's also interesting because it's also one of the genes that for adaptation to altitude. As conclusions, we have developed a novel approach to identify gene-environment interaction in a large number of environmental conditions. Most of the variants are shared across many conditions. This is also something similar that happens on GTEx, that you have a lot of sharing across tissues. Genes with a higher change in gene expression are more likely to show treatment-only or control-only allele-specific expression. And we have identified convincing examples of condition-specific regulatory variants. And what we would like to do next is then to use this type of information to also inform genome-wide association studies. And finally, the acknowledgements, as I mentioned at the beginning, this was joint work with Francesca Luca at Wayne State and William Wan at the University of Michigan. Greg Moyer-Brilliant, who will present later in this session, did most of the gene expression analysis. Chris Harvey did developed Quasar and did the ASC analysis. Cindy is doing some validations, and she will present a poster on unrelated work. And finally, knowledge funding, and we are hiring postdocs. So Detroit is a nice place, despite everything that you might have heard. So if not, you don't believe me, come and visit, and I'm sure you will like. I might be able to convince you. Any questions?
I think we have time for one question. Anyone has any questions? Nancy.
So this apparently shared architecture is sort of ubiquitously seen. Everybody puts in their papers, the ones that look very context specific, but the vast majority of what's discovered looks shared. Do you think that's real, or do you think that the cellular architecture that might drive real differences is so well represented in what we say is the control sample?
Yeah.
You know, whether that's driven by pH or other, you know, whatever it's really driven by at the cellular level, that we can't really see the context as well as we think we are, or not? I mean, so is there a lot more real context specificity, do you think, than what we can actually measure?
That's a great question, and I would like to know the answer to. I think that a lot of the things could be maybe less active on the baseline, and then and basically you're still be able to detect. I mean, one thing that it may be interesting too is that knowing also that the gene responds and that having an EQTL may be enough, but that will be something that probably we have to investigate.
Okay, let's thank the speaker again.